Welcome again to this week's issue of Video Table of Contents from the editor's desk. We have an exciting lineup of studies for you to peruse, so let's get started.
In a novel study by Datla et al., the role of polymerase delta interacting protein 2, or POLDIP2, was studied in relation to movement and migration of smooth muscle cells. Using an overexpression paradigm, these authors showed that overexpression of POLDIP2 impairs migration and dissolution of adhesion of vascular smooth muscle cells. The data also show that POLDIP2 is an important regulator for migration via the NOX4 Rho-A FAC pathway.
Keenast et al. Used the technique of growing cardiomyocyte monolayers on microelectrode arrays to study the effects of changes in propagation directions on field potentials and conduction velocities. The authors found temperature-dependent changes in the dominant pacemaker potential and conduction velocity variations. These data have implications for understanding field potential spread in electrophysiological experiments. Thomas and co-workers investigated the role of myocardial NF-kappa B in diabetes. In this study, transgenic mice overexpressing a mutant I-kappa B isoform in the heart prevented activation of NF-kappa B signaling. The authors examined calcium signaling and the involvement of the renin-angiotensin system in diabetic, transgenic, and wild-type mice. The data show that inhibition of NF-kappa B signaling in the diabetic heart prevents cardiac dysfunction by preserving calcium handling and inhibition of the renin-angiotensin system. Thank you again for joining me on this edition of Table of Contents Video Highlights. We will see you again in 2 weeks, but keep sending us your best papers. Remember, AJP-Heart and Circ impacts cardiovascular science in a major way.